So the next talk is a remote presentation.
Hello, everyone. Thank you so much for being here. My name is Rishabh, and I'm a grad student at Yale. Today, I'm going to talk to you about RNA-FrameFlow, a generative model for de novo 3D RNA backbone design. It's something that I've been working on with amazing company for the better half of 1 year. This was for my undergrad honors thesis, and I had the chance to work with all of these amazing people from the Cambridge Computer Laboratory in Pietro Leo's group. I think there's a Q&A segment at the end, and I'd love to answer questions along the way, if any. Yeah, let's get started. So just a fresher on nucleic acids, or specifically ribonucleic acids, RNA. They're super important. They facilitate protein synthesis, gene regulation. They essentially make you, you. And over the past few years, there's been a lot of interest in RNA biomedicine. We've heard a lot of action in terms of CRISPR and, because of the pandemic, mRNA vaccines and a whole bunch of other related therapeutics. Um, what's interesting is that from an AI standpoint, most of these RNAs that are important, they have some sort of important structure. For example, riboswitches, ribozymes, they're just examples of many types of ribonucleic acids that can be sort of hijacked or modified to act as really important biomedicine in the body. So essentially, what our goal is here is we want to build tools that can design custom new novel RNA and eventually figure out what function it would serve in the broad scheme. So I think in terms of generative modeling for structural bio problems, there's a lot of emphasis on proteins. It's seeing a lot of action. But RNA is pretty similar and pretty complex as well in terms of being a modeling problem. We know proteins to have 4 backbone atoms. The RNA nucleotide has 13 backbone atoms. So in terms of complexity, that's slightly higher in terms of modeling. Proteins have 20 amino acids and RNA have 4 bases, A, U, C, G. And protein structure, the tertiary structure that uh, drives protein folding or protein structure, it's driven by H bonding, hydrogen bonding along the backbone. But in RNA, it's between the bases themselves. And this gives rise to really important or interesting phenomena like base pairing and base stacking that give you that really globular 3D shape. So this work, we wanted to present a geometric generative model for this task of 3D RNA backbone design. And as you can see, this graphic, we want to go from complete noise or like nothing, and we want to generate these RNA structures that are novel and nice and have desirable properties if you want to use them for something else. Just to get the crowd excited, these are some samples generated by our model, and I'll eventually get to how we were able to generate these and some other topics worth considering in this project. So I think since this is mostly a machine learning slash bio audience, I think data is such a huge important factor. So for us, we wanted to find a rich enough source of RNA structures, 3D RNA structures, and we were able to come across RNA Solo. It's this database of cleaned PDB structures, and it features these structures from several sources. They could be solo isolated RNAs, they could come from complexes, and altogether, in total, there's about 15,000. And when you do the filtering and when you actually look at things that would be considered useful or nice to generate, we sort of filter that even more and look at any RNA between 50 nucleotides and like 250 nucleotides. And that really gives us a really diverse set of RNA structures of varying length. It gave us about 3,000 structures that we were able to play with. So next, an important task here is I mentioned that RNA backbones, every nucleotide has 13 atoms. Modeling each and every of those 13 atoms would be very computationally expensive. So we wanted to find alternative methods to be able to represent them easily so that in terms of modeling or generation, it's simple. And maybe you could retrieve the full atomic scale, atomic resolution structure easily as well. So in sort of protein land, there's a lot of excitement about using these things called frames, how you could parameterize the backbone using just a rotation and a translation from some global origin. And you can think of these frames as representing each nucleotide. And the RNA structure or the protein structure could be a collection of these frames arranged in 3D space. So that's what we're dealing with. And we essentially take these three anchor atoms, C4 prime, C3 prime, O4 prime, and we perform the Gram-Schmidt operation to get this frame T, which comprises or encapsulates this rotation and relative rotation and absolute position in 3D space. And of course, you might be wondering, hey, there's 10 other atoms you might be forgetting about. How do we get that back? So for us, we auto-regressively place them in. We fill them in one by one using these final output frames after the model has done its thing. And we're able to get back all these atoms by modeling it as these angles, these bond angles that you can easily predict using some sort of MLP or auxiliary network and just sort of place them for each nucleotide in the backbone. So sort of the main workhorse of the engine of our generative model, it relies on this relatively recent paradigm called flow matching, and this specific variant called SE3 flow matching. SE3, it's essentially the group of all rotations and translations, which basically is the sort of set of rotations and translations that you can sample from to create these diverse backbones or to place these atoms or nucleotides in 3D space. So we do flow matching on this set, and we want to generate a set of these rotations, translations. We want to predict those angles and get the other remaining atoms, and that allows us to recapitulate or retrieve the full atomic resolution structure. So this is just a TLDR on flow matching. It was devised by these two papers specifically in the context of protein design or just relying on things that are from SE3 or any sort of manifold. So it starts off with rotations and translations. You can consider them separately. The math works out really nicely, and you can sort of independently do or sample along the rotations and translations, and that allows you to get those frames. And essentially what flow matching does is it starts off with random noise, both the translation and the rotation aspect of the frames, the nucleotide frames. And essentially we take these steps, these T number of steps, and it allows us to update the atomic positions or these frame positions in 3D space. So you can think of it as starting off with this really, this gas of frames, this really just random point cloud. And eventually you iterate and refine this structure. You update these frames, and these frames are eventually converted to all atom coordinates. And then you finally get the very nice looking RNA structure over here. Hannes Stark has a very interesting flow matching visualization in his video from his recent paper. Yeah, I'm not entirely sure of forgot the name, but it has a very nice visualization. Maybe I'll include it into my slides later when it's shared publicly. So essentially we're doing the same thing. And in this step where we're denoising or we're updating the frames, it passes through this neural network, which is the denoiser neural network. So between any two intermediate states, we pass it through this denoiser multiple times, and you can think of it as a refinement of frames. This denoiser that performs these updates, it is based on the AlphaFold 2 structure module. It contains very nice components like invariant point attention that allows you to do geometric modeling, geometric attention in a very nice way. And it allows allows the network to be expressive enough to really learn the distribution of 3D RNA backbone structures. So this is just a quick animation of what exactly is happening. You can see that it starts off from this random gas, and eventually you iterate and you update these frame positions. Each of these bubbles that you see, so the color is sort of irrelevant, but you can see these bubbles, and each bubble is a frame in itself. And you can see how these frames are arranged and updated so that they've come to form this very nice looking RNA backbone. So I think now that I've talked about the generation aspect, how exactly are we evaluating it? We were hoping to do something similar to the protein design literature. We wanted some local metrics to show that, okay, if you sort of zoom in, it looks like RNA, not sort of gibberish. At the same time, we wanted to see, okay, the RNA that we're generating, how does it hold up against RNA that exists in the wild? Is it good enough, essentially? So we've come up with a few local and global evaluation metrics that help us see or test, evaluate our model. So evaluation, local evaluation, we hope to retrieve sort of physical quantities, physical measurements like atomic distance, bond angles, dihedrals. And I guess coming from a computer science background, the Earth mover's distance is a very nice measure of similarity. So we're essentially computing this for every single structure in the training data set. And we do the same for our generated RNA. We generate a bunch of them, and we see whether these measurements match, whether we're seeing similarities in the distributions. Next, we want to globally evaluate our RNA, and this is very similar to what we've seen in the protein design literature. So we want to check some sort of validity. So if I generate a backbone, is there an AUCG sequence that can eventually fold into our backbone? This is also called self-consistency. And in the protein design literature, sometimes it's called designability. And for us, we measure this using self-consistency TM scores, self-consistency RMSD, and self-consistency global distance test, which is used in the protein design literature very commonly. Next, we want to check diversity of all the samples that are so-called valid. How different are they from each other? Are they diverse, or are you seeing some sort of sad mode collapse issues where it's just generating one single type of RNA? And of course, we want to check novelty compared to everything that's out there in RNA solo or in the wild. What's the new stuff that we're cooking up in our generative model? So here's a note, a sort of word of caution that unlike proteins that have gone through experimental validation, there's no real notion of designability for RNA yet. We haven't really seen AI designed RNA go into wet labs, and at least publicly, we haven't seen it being done. So that's why we call it validity or self-consistency. And we use a TM score threshold of 0.45. And the medicinal chemistry literature says that RNA with a TM score above 0.45, they sort of have the same global fold, given how flexible these RNA are in general. So let's get started with some very interesting results that we've seen. Right off the bat, we can see that our generative model can successfully retrieve these physical or geometric quantities. You can see that we match the training distributions between the data set and our generated samples in terms of bond distances, angles, and dihedrals. And if you compute the equivalent of the Ramachandran plot for RNA, which you can think of it as, since there's phi and psi for protein backbones, you can create pseudo bonds between those 13 atoms in a nucleotide, or pseudo bonds between these nucleotides, and compute similar angles, theta and I think that's nu. Yeah, theta and nu. And it allows you to almost relatively retrieve the underlying angular distributions. And I think in terms of global metrics, we hope to show that our method, if you compare it to existing baselines like MMDiff, which also came out recently, we're able to get a validity score of 40%. And MMDiff being a diffusion model, or like a vanilla diffusion model, we weren't able to really squeeze enough performance out of it, which is why we left it blank. We weren't able to get any valid structures from this model, even after retraining. So I think this is one of the key results that we'd like to highlight, that in terms of validity, in terms of self-consistency, TM score, SCTM, we're achieving about 41%. And you see similar very nice values for diversity and novelty. We present further ablations in our paper, so definitely do check that out. We try out different settings for RNA-FrameFlow. We play around with the hyperparameters, and we try to give you the best model, which is open source, by the way, and I'll get to that soon. Next, we want to look at how realistic or how valid these generated backbones are. And you can see that simply because of the structural diversity or the current structural diversity in RNA solo, you get this sort of bias, length bias towards certain sequence lengths like 70, 80, 120. Essentially, we want to be in this zone here where the self-consistency score is very high, and you see low PDBTM, which means it's pretty novel. You don't really see similar structures in the PDB. So how exactly are we measuring this SETM? I want to go back to this sort of global flow chart that we have. You have our generated backbone, and we pass it through this pipeline where we inverse fold the backbone to get the AUCG sequence. Then we pass it through this model called row fold, also called E2E fold recently. There was a name change. And that allows you to do structure prediction on top of that generated sequence. And then you would sort of superimpose and RMSD align these or TM score align these generated backbone and predicted backbone. And if there's a high TM score, you would say that, hey, and if it's above 0.45, you would consider this generated backbone as valid. So that's how we compute SETM score using the grenade and row fold pipeline. So yeah, TLDR, we can see that our model is able to generate some valid structures, some novel structures. But of course, there's still a long way to go. And it's limited even by the structure predictor's performance. And in our paper, in fact, we highlight that row fold may not be the best structure predictor out there. So there's definitely a lot of work that needs to be done. So in our current model, we sort of make a trade-off between novelty and validity. We've observed that valid structures, simply because of the diversity in RNA solo, the valid structures, they look like they're from the PDB, or they look pretty similar. They have very relatively high PDB-DM scores. But then we can see on the other hand that the novel structures, the ones that our model is able to uniquely generate, row fold is not able to, the grenade and row fold pipeline isn't able to retrieve it properly, which might be an issue given the current state of tools for RNA design. So again, these are just select samples, and with these metrics, you can see that we've, for certain sizes, certain sequence lengths, we see that trade-off between validity and novelty. Otherwise, the scores usually are pretty decent in terms of the RNA looking locally realistic or locally plausible. And that's one of our strengths in the paper that we hope to highlight. Of course, there's the other side of the coin where our model still is prone to some limitations. We end up getting RNA that where the strands of the RNA are sort of entangled with each other, or they might have these sort of stray loops in the middle of nowhere. That means the frames corresponding to the nucleotides along this stray strand, it was just generated and placed over here, and it has no bearing on any of these, the broader structure of RNA. And you can see that it also generates these really, really highly coiled RNA structures, which could be an artifact of the structural diversity in RNA solo, even with a bit of cleaning and processing. So we present methods in the paper that curb and control these issues. So we include data augmentations of different types, structural clustering, length-based clustering that allows, or length-based sampling, all these tiny tricks that we've tried to really squeeze the juice and really get the most out of RNA-FrameFlow. And again, that's all open source for everyone to try out. And with that, that brings me to the end of the presentation. Thank you so much for your attention. If I had to really conclude, we've seen that these current generation of protein design tools, they are expressive in recapitulating, retrieving RNA structural biology to a certain extent. We've seen that lack of data diversity, it's a broader problem in AI for science, but specifically for these very niche, relatively niche biomolecules that are not proteins like RNA and DNA. And we hope that that's something that the community gets to work on together. So over here, we've just sort of randomly taken frames and we sort of update them to create these very nice looking 3D structures. But then now the next steps would be being more principled, sort of sticking to the science, and trying to incorporate notions of base pairing and base stacking to ensure that the diffusion process or the iteration, the flow matching, it's a lot more realistic, and you get structures that don't face these limitations like coils and stray strands. You see something that would mimic something you'd see in the wild. Of course, we want to bring in improved evaluation metrics, maybe bring in secondary structure-based evaluation. So far, we've been evaluating the 3D structure, but I'm sure there's a bunch of exciting things that can be done in really showing that the generated RNA are both locally and globally realistic. And of course, I think naturally you'd want to extend this to more conditional design settings. So that would come in the form of aptamer design, RNA aptamer design, where you have some sort of target protein and you want to design a binder RNA that has high affinity, has nice properties. And of course, now given the recent surge of these biological LLMs, very recently there was an RNA language model called Rhinalmo that came out of a lab in Singapore, and that's really exciting because it gives us nice sequence embeddings to play with, and those would be natural extensions to this project. And with that, yep, thank you. I'm open to any questions. And one last thing before I end, you can check out our paper on arXiv over here, and you can check out our GitHub. I promise our documentation is pretty good. So the paper's over here, and our code base is, yeah, it's fully documented for you to hit the ground running. And with that, yeah, thank you so much.
Thank you so much. So we have time for maybe 2 quick questions.
I had a question about how you parameterize the parameterize the torsions of. Yes, exactly. So do you just have a prediction head that predicts those torsions from the diffuse structure at the end? Is that right?
Yes. So once the frame, the central frame, these anchor atoms go through the whole diffusion process, we have an auxiliary network, much like RF diffusion, or even frame diff, that predicts these individual angles, 8 to be precise, and then we'd auto-regressively place these atoms based on these angles.
Got it. Cool.
Does that clarify?
Yeah, and then did you look at diffusing over those torsions as well during, or I guess, I don't know what the analogous word to diffusing is for flow matching, but did you try it all flowing over those torsions as well?
We actually did, and we've come to find that using an auxiliary network is a lot better because in terms of the diffusion, sometimes you run into these issues like the lever arm effect, where maybe if you update these angles a bit by bit, you could have this OP1P phosphorus bond sort of pointing here, right? So we found that MLPs are a lot simpler, a lot more robust to these sort of global angle orientations, and it was just able to recapitulate the angular distribution a lot better. We didn't really study the like how close or how similar the MLP distribution of angles was to the ground truth angles, but I'm sure that an MLP would suffice here, much like is as similarly used in these protein design models as well. But you can definitely also do angular flow matching on like the hypertorus or something like that, and it would get you these very nice angles. But maybe it might take a bit more optimization, I think.
All right. Thank you. We're going to move on to the next talk.
All right. Thank you, everyone.